Great, thanks very much for coming, everyone. So this is a slide that by now you're no doubt completely familiar with. It's just a very simple kind of schematic of relationships between living jaw vertebrates. Basically, we've got two groups. We've got the cartilaginous fishes. These are the sharks, rays, and chimeras, and there are about 1,000 living species of them. And then we have bony fishes. And the bony fishes can be divided into two main groups. The ray fin fishes, these are things that you could say would be seen on a dinner plate or in an aquarium. So things like tuna and goldfish and cod and seahorses, that kind of thing. And there are about 30,000 living species of them. And then we have the lobe fin fishes. And these are things like coelacanths and lungfish, but also all land tetrapods and other tetrapods. So humans and dinosaurs and snakes and all that kind of thing. And jawed vertebrates, of the jawed vertebrates, the bony fish are the most dominant living group. And the ray fins, the actinopterygians, actually account for over half of living vertebrate diversity. So the ray fin fishes are the actinopterygians, so named because they have particular types of rays, fins in their rays. Rays in their fins. But despite this staggering modern diversity, we know very, very little about the early evolution of the group. So they diverged from Sarcopterygians about 425 million years ago. But if we look at the early fossils of the group, we have no unequivocal Devonian actinopes, Silurian actinopes, And in the Devonian, things get a bit better. We have about 20 or 15 actinopes, but we don't really know very much about them. Most of them are only known from the dermal skeletons. These types of fish have two skeletons. They have an external bony skeleton, which is primarily for defense or protection, and then they have an internal endoskeleton. And the endoskeleton is really useful because it's very complex, and it includes a thing called the brain case, which sits in the skull and houses the brain and sensory organs. And the brain case is common to all vertebrate groups, so you can compare it between different taxa and different groups. But it's also very complex, so it's really useful for helping to tease out relationships between these different taxa. The things I've highlighted, we only have very good endoskeletal descriptions for Mimipiscus and Moitomazia in the blue. Mimipiscus is this thing here, so this is its brain case, and this is a reconstruction by Brian Chu. We have partial brain case data for Chirolepis, Halquillepis, and Gogo sardina, but either it's not very well described or it's not very well preserved. So what has basically happened is that Mimipiscus has come to be the exemplar of what Devonian actinopes look like. We don't know whether that's true or not because we have no data to compare it with. So at the moment, it's just taken as read as Mimipiscus is representative, and it's typically what is used as the kind of definitive ray fin in various different analyses. So a couple of years ago, Laurent Darras came up to me with this fossil and said, I don't really know what it is, it's some kind of actinope, can you do anything with it? So the scale bar at the bottom is 1 centimetre, so it's not a very big fossil, it's in lots of different pieces, it doesn't look very exciting, especially if you don't work on fish. And it's from the Upper Devonian of northern France. So we thought, we'll stick it in the CT scanner, see what it looks like, see how it is. This fossil is basically amazing. It's an articulated head of this Devonian actinope. It's got the dermal bones in the skull roof and the cheek. This is the jaws here, so you've got the lower jaw and the upper jaw. You've got a brain case in there. This is where the eye would sit, so anterior is to the left. But perhaps most interestingly of all, it's got this gill skeleton, which is this kind of yellowy green thing that you see. And we only have one described actinope gill skeleton from the Devonian, again from Mimipiscus. And then we have some from the Triassic. But this, again, not very much data there at all. So I'll run through a couple of the actinope characters. It's got this kind of what we call a ganoid ornament on its skull roof, so we know immediately that it's an actinope fish. It's got an enclosed dorsal aorta, so this is the brain case. We're looking at the ventral surface, and this is the main blood vessel that would have supplied the head, the eyes, the brain, everything. So it's quite an important character. But we can also see that it has some very primitive characters. So this is the jugal. The eye would sit kind of here, and the jugal's got this thing called this kind of notch in it, this orbital notch. And that's only seen in one other taxon, in Chirolepis, which is resolved as the most primitive actinope. So we know that this thing is quite primitive. It's also got this hole in its palatal quadrate, so that's the kind of internal cheekbone. So it's very definitely an actinope, and very definitely a fairly primitive actinope. As I said, it's got this complete brain case, which is really useful for understanding relationships. I could talk about the brain case for 15 minutes, but I'm sure that no one cares and everyone wants coffee, so I'll spin through it. But basically, these circles represent the eyes, and this is in dorsal view, and this is in ventral view. So as I said before, we've got this enclosed aortic canal, we've got very big craniospinal processes. The portion of the brain on which what's called the lateral dorsal aorta is very long, and the spiracular groove is very wide. We can have a look in this is lateral view, this is anterior, this is posterior, and again we've got this very wide spiracular groove. There's this little thing here called the ventral aortic fissure, which again was previously thought to only be seen in a couple of animals, but it turns out this looks like it's a fairly common feature. We can also have a look at the endocast. So for those of you who don't know, the endocast is an infilling of the internal brain cavity, the internal brain case cavity. So it's a space that would have housed the brain, the sensory organs, and kind of the fluid surrounding the brain. And in early actinopts, the endocast and the brain are a very good fit. So we can use the endocast to look at brain evolution in these animals. So this is in dorsal view, this is the top of it, this is the side of it. A couple of features, we've got these optic lobes, and you can actually see here, this is the optic nerve, so that's what would have innervated the eye. So the optic lobes aren't very large, which again is quite a primitive character. And this is the bony labyrinth, so the equivalent of our inner ear, and it sits fairly low down, which I'll talk about a bit later. But as I said earlier, the gill arches are really one of the most interesting things about this, to have a complete and nearly completely articulated set. For people, again, who don't know much about gill arches, this is a kind of schematic of a tuner. We've got this median element here. We've got a series of repeated dorsal and ventral elements. And again, this is in dorsal view, so you've got your median element here, some ventral and some dorsal series. And I'm just going to run through the gill skeleton very quickly. This is the French fish on the side here that I'm talking about. This is an animal called Palaeontological. So Palaeontological is often resolved as the most primitive living actinopt. And I've kind of coloured the elements the same so that you can have a look at the gill skeleton as I build it up. But basically this light purple thing is the brain case, and then these are different gill arches. So again, in both taxa, this is Palaeontological, this is the fossil. We have this median ventral element called the base branchial. Then we can build up all of the hyoid arch and the various gill skeletons. So we've got these ventral series and a dorsal series of ossifications, and then various other bones that are only of interest to me, so I won't talk about them too much. But we can build up a picture of what the gill skeleton looks like reconstructed. And we see all the different elements there, and this fits in really nicely with the kind of results that Alain Bridel put forward in a Nature paper this year about the gill skeleton and chondrichthyans, and it feeds really nicely into that story of, the kind of primitive gill structure in bony fishes, and chondrichthyans is quite bony fish-like. So this is sort of the plesiomorphic living jawed vertebrate gill configuration. So, I'm talking about this fish. Who really cares about it apart from me? Well, it's really significant. As I said before, the brain cases are really important in trying to understand relationships between these animals. This is the group that today accounts for over half of all living vertebrate diversity, and we don't know anything about their evolutionary origins back at the Devonian. So in having this brain case, I'm calling it provisionally Reinerius after Dorothy Rayner, who was a paleontologist in the 1950s. We've got this kind of really good level of preservation that we can use directly to compare to Mimipiscus, which is what we've used at the moment for kind of representative actinopt condition. First of all, we'll stick it in the phylogenetic analysis. So this is edited from the matrix of Chi 2011 with an expanded character and taxon set. And Reinerius, it's not really interesting to anyone apart from me, but it plots out here, so it's fairly primitive. It plots out with an animal called Moitomycia, and then these are the more kind of carboniferous grouping of animals. Because I've expanded the character set, we can also use it to look at relationships amongst early total group osteichthyans. So Ligula lepus is coming out as a stem osteichthyon, and Mumania and Dialipina are coming out as a stem actinopterygian, which is quite an interesting result, but it seems to be borne out by the characters that we're coming up with now. But I mean, really the most important thing of this is that we can compare it to mimipiscids. So I've kind of aligned the brain cases on common points of anatomy. So we've got the ethmoid articulation of the nose up at the top, this thing called the ventriloquial fissure, and then the back of the brain case. And we can see that overall they look quite similar. They've got the same kind of bones in the same kind of places. But if we pick a few more landmarks, so the top of the postorbital process, again the orbit would sit here and here, the exit of the high root of the branchial artery and the front of the aortic canal, we can see that the proportions between the two are quite different. So mimipiscus has got this elongate, very narrow occipital region, whereas it's much shorter in Ranierius. The portion of the brain case in which the lateral dorsal aorta are carried is much longer in Ranierius, and the postorbital process and lateral commissure is much narrower in mimipiscus. So we can then go and look at some other animals that have recently been described, such as Chirolepis, which is currently being submitted, which I've just described. And we can look at things that have been figured but not necessarily described. And we see that actually Ranierius seems to be much more representative of other early actinopes. So many of the features that we might have thought were primitive in mimipiscus appear to be apomorphic. So it's a sort of cautionary tale that if you only have one data point, you can't say that that one data point is representative of everything. You need at least two. So you need to find more fossils, basically. Keep looking. But this also feeds really nicely into the picture of endoclasts of brain evolution that we've been building up in recent years. So the optic lobes, which I talked about before, these things here, they innervate the eyes. In sarcopterygian outgroups, the optic lobes are really small, and they're a little bit bigger in the earliest rayfins, but they get much bigger in Carboniferous actinopes. And also we can see that the bony labyrinth, the inner ear, in sarcopterygians it's quite dorsally positioned. I mean, it becomes more ventrally positioned as you go through actinope evolution. Final point, I mentioned about the brain cases, I've been banging on about them. How important are they really? Well, this is, again, just showing again my phylogenetic analysis with all the brain case characters in it. What happens if you take them out? It collapses. So there has previously been an argument by Gardner and Schaefer in 1989 that brain cases are too conservative to be of any use in understanding relationships between these. And as a result, people haven't possibly gone into them in as much detail as they could have. They're seen as interesting things, but they may not have been mined for characters as extensively as they could have been. But you can see if we take the characters out, the entire tree collapses. So we have a little bit of resolution where these things, where Chirolepis is coming out as more primitive, but really most of the resolution has been lost. So it's a really interesting and really important piece of anatomy that needs to be mined, and we need to look into it for more characters to understand the origins of this kind of really dominant modern group. Thanks very much.